Again, I'm Lori Pavarnik at the University of Rhode Island, and because the moderator of this particular session could not make it, David asked me to take over, although now that he has a Lifetime Achievement Award, I really do think he should be doing this. So we have a great session. It's biotechnology and aquaculture. We have two wonderful speakers. The first speaker is Ronald Stotish. He's the CEO and president of Biotechnology, and he will talk of AquAdvantage salmon, potential for sustainable salmon aquaculture. And both gentlemen who will be talking in this session will sort of introduce themselves. So, oh, I didn't even have anything to drink. So there you go. Sorry. I knew you were here someplace.
Thank you for that wonderful introduction. I did agree to introduce myself. I will tell you a short story. My youngest son is 33 years old. When he was about 10, he brought one of his little friends home. And the little friend said, "What does your daddy do?" And my son looked at me, and then he looked at his friend, and he said, "My father used to be a scientist." And the reason he thought that was that when he was younger, I used to take him into the lab, and I used to let him pour plates, and I used to let him count bacterial colonies. And a little bit later, we used to go to the experimental farm. He could play with the baby pigs and so forth. And as my career progressed, I had an office, and I had a staff, had secretaries, and I still had the same scientific responsibilities. But in Tim's mind, once I left the lab and took the white coat off, I was no longer a scientist. And I have to tell you, it was like a stake through my heart when that little boy said that. And I've had the opportunity to embarrass him on several occasions when he's been in the audience by repeating that story. I'm essentially a product of the pharmaceutical industry. I spent probably more than 35 years from discovery of new products in human and animal health to new product development, regulatory affairs, and research management. I've had global responsibility, basically managed global animal health businesses. After that 35 years, I decided to give biotech a try, and I've spent about another 15 years in biotech, most recently with AquaBounty. The reason I joined AquaBounty was, at the time, the AquAdvantage fish had been in existence for some years. The AquaBounty board felt that they needed to bring someone in who had product development and regulatory experience, and I had registered over 100 products around the world. And so they asked me to join the company. And the only reason I joined was I felt that this was a unique opportunity, and my hope was that we would gain the first FDA approval for a genetically modified animal. And at the time, I wasn't entirely sure that it was possible to get an FDA approval for a genetically modified animal. Fortunately, it worked out. I won't tell you it was easy. It was, in many respects, frustrating, aggravating, but at the end of the day, also very gratifying. And along the way, I made a lot of friends. I learned a lot, and I've had to work and had the opportunity to work with some very wonderful people. So what I'm going to tell you today is the story of the AquAdvantage salmon. Most recently, the regulatory process and approval. And I'm going to touch on some of the issues that Jack Bobo introduced in terms of the interaction of science policy, regulatory policy, the interaction of consumer perception and production, aquaculture, the importance of aquaculture to our developing world, and the opportunities that we have as we go forward. This slide overstates it a little bit. It says everybody loves salmon. I know everybody doesn't love salmon, but a lot of us do. Salmon's the second most popular seafood in the United States, right after shrimp. It's the most intensively cultivated fish in the world. And I think as a way of introduction, I'm going to talk to you a little bit about salmon, because although we all think we know a lot about salmon, I suspect that a large percentage of the audience really doesn't understand the global salmon industry. Jack talked about this a little bit, and I have a few slides that deal with this. The wild capture fisheries has been flat at about 100 million tons for about the last 30 years. That's another way of saying, even in a well-managed environment, there's no more to be gained from the wild-caught fisheries. Aquaculture has been growing at roughly 7% per annum for about the last 15 years, and probably over the next 10 or 20 years will grow another 20 or 30%. Most of the new growth of seafood is coming from the aquaculture segment. I like to use this statement because it refers to the milestone, and the milestone is that about 50% of the world's seafood for human consumption was to be supplied by aquaculture, and this was in 2009. We've exceeded that now. It's over 50% of the seafood that's consumed by humans worldwide comes from aquaculture, partially because the wild-caught fisheries have been overexploited, overfished, and we've gotten very efficient at using large factory ships to go out and harvest huge quantities of fish. The problem was, even in well-managed fisheries, there's no more to be had. So we can maybe stay even with what we have, but we're not likely to improve the wild stocks. On the other hand, and this is a very plain Jane slide compared to what Jack showed, the world population is continuing to increase, and I'm not alarmed by that. Jack gave you the statistics this morning and the demographics. One thing that's important, we will have to feed those additional 2 million people by 2050. We're already at 7 billion people, soon to be 9 billion people. And what Jack didn't mention is a lot of those new people are in emerging middle classes in developing countries. And they're demanding higher quality, more interesting diets, including higher protein, for instance, that come from either livestock or seafood. The global salmon industry is a $10 billion industry today. It is exclusively, almost exclusively, a cultivated species. If you're eating a wild salmon, you probably caught it yourself. Wild-caught salmon represents less than 2% of the consumed salmon worldwide. Everything else is farm-raised. That's Atlantic salmon, and I should make that distinction. For Pacific salmon, there are fisheries. They're reasonably well-managed fisheries. There's a technique called ranching, where the young fish are raised in hatcheries, for instance, in Alaska, and then released at about 100 to 200 grams, and a couple of years later they catch those fish when they mature and come back to where they were released. Those are Pacific salmon, and it's a number of different species. There are 7 significant commercial species, but there is a real distinction between Pacific salmon and Atlantic salmon. And a simple way to keep them straight is that's Atlantic salmon. Pacific salmon is a lot more like trout in terms of texture, appearance, and so forth. Both are valuable food commodities. Both are highly desirable products. Both are very rich in high-quality protein, omega-3 fatty acids. The U.S. imports roughly 300,000 tons, or over 300,000 tons, of Atlantic salmon every year. And from now on, I'm going to be talking mostly about Atlantic salmon until I come back at the end, and we'll talk a little bit about Pacifics. But the U.S. imports over 300,000 tons of Atlantic salmon. We produce very little. There's a little produced in the state of Maine. But the preponderance of the salmon that we consume comes from places like Norway, Chile, Canada, Faroe Islands, Scotland, the UK, Ireland. And you can see the country breakdown in that somewhat complicated slide. That salmon has a value of over $2 billion per year. I regard it as somewhat of an issue of national pride that we don't produce more salmon here in the United States. We have the technology, we have the genetics, we have the ability. In fact, in many of those countries, they're using American technology and genetics to produce those salmon. But we've chosen not to for some of the reasons that Jack mentioned in his analogy with the EU exporting their agricultural production capacity to Brazil. And I think, frankly, we should reconsider that because there is an opportunity for economic development, job creation, technology, and putting food closer to consumers by producing these fish here in the United States. To underline the importance of aquaculture, in 2000, the Atlantic salmon was declared an endangered species off the coast of Maine, in the state of Maine. That was declared an endangered species because it was overfished, habitat was destroyed, and a whole lot of other reasons, most of which were man-made. There are people who say if only we managed the fisheries better, we could restore those wild populations. And there are wild Atlantic salmon in the Great Lakes, for instance, and there are wild salmon off the coast of Maine. There are wild salmon off the coast of Nova Scotia and in the Atlantic provinces and so forth. But over the past 30 years, there have been no less than 70 attempts to reintroduce those stocks and to generate viable reproducing populations, all of which have failed. They failed because we've destroyed the habitat, we've developed the coastal waterways, or other reasons. So it's not a gloom and doom story. There are wild salmon. Compared to what we're consuming, there just aren't enough of them to support the consumption of Americans. So where do the salmon come from? They come from sea cage operations, of which this is an example. These are cages that are moored off coastal waterways. They're tended by boats that bring out feed and managers that manage them. The fish are put in these cages when they're anywhere between 100 and 500 grams. They're raised to market weight, which is generally between 4 and 6 kilograms. And then they're harvested in those boats, they're killed, and they're taken to processing plants. It occurs in areas that have long coastlines of cold water, places like the North Atlantic, around Norway, Faroe Islands, UK, Scotland, Iceland, the Maritimes in Canada, and in the South Pacific, where the Chileans have created an Atlantic salmon industry because they too have long coastlines with protected inlets suitable for anchoring these cages. There is some salmon production, Atlantic salmon production, off the coast of British Columbia. They're not well received by native peoples or by Alaskans, but there is some cultivation there as well. Alaska has laws preventing aquaculture in Alaska, prohibiting aquaculture in Alaska, among many laws, interesting laws that they have in the state of Alaska. Sea farming is a highly developed high-tech industry, and it produces, as I said, over $10 billion worth of salmon every year. It's a very successful industry, but when you think about it, those cages moored off these coastal waterways, first of all, if you medicate the animals, because you have outbreaks of bacterial infections or what are called sea lice, which are small crustaceans which attach themselves to salmon and cause mortality and morbidity. Seals get into these. To a seal, this looks like a deli or a shark. They get in and tear the nets, release the salmon. The salmon get out, have the potential to mix in the local ecosystem, potentially interacting with other species. You have jellyfish and algal blooms. The industry off the coast of Chile has had a sequence of natural disasters that rivals Job's trials of biblical origins. They've had volcanoes. They've had 2 rounds of ISA outbreaks. They've had algal blooms that destroyed 40% of their productive capacity. And their industry is roughly at about 50% of their capacity of 10 years ago. All based on natural disasters or management issues associated with the cultivation of Atlantic salmon. The weather, you can imagine what a hurricane or a typhoon can do to these anchored sea cages. And the opportunities for improving the transportation footprint. Those locations that I've described to you are all at great distances from New York or Miami. So you basically are flying, if you're flying fresh fillets, you're flying fresh salmon in 747s from the south of Chile, 3,600 nautical miles, or from Oslo, Norway, 3,200 nautical miles to the New York or Miami markets. The carbon footprint associated with that is huge. The reason they're in those remote locations is that's where those coastal formations are favorable and the water is cold. There has to be a better way to produce salmon. So let's talk a little bit about sustainability, because that was a major focus of your conference. This goes back to the World Earth Summit in 1992. Sustainability reminds me of the old joke that I certainly don't know what it is, but I'll know it when I see it. And everyone and every major organization has some sort of a sustainability platform, policy, or statement right now. It's a little bit like natural. It's a little bit like a lot of other stuff. And without sounding too cynical, I think there is an opportunity to talk about the realities of sustainability. There's economic, social, and environmental sustainability. I have a couple of quotes here from Hilborn at Washington and talking about the Global Sustainable Seafood Initiative. This is a process for responsible certification. And to be honest, it's a good program. This is a post from Ray Hilborn from the University of Washington, and he talks about this. And this is after the World Wildlife Foundation backed out of a certification program that they were working with. And World Wildlife is actually one of the better NGOs. They're good people, they have high standards, and they're certainly well-intentioned and well-meaning. But at the end of a process, Monterey Bay, Seafood Watch, and World Wildlife backed out of a plan on the GSSI. And Hilborn makes an interesting point here. The underlining is mine, not his. I should make that clear. But this is his quote. "Not too many people buy their fish based on sustainability readings." I know that, and I think you know that. When my daughter or my wife go into a market, sustainability is not really on the table. They look at quality, they look at price. They may have a social consciousness about sustainability, but it's not the first thing they think about. But if you're a big company, whether you're a Whole Foods, Costco, Safeway, you don't want people picketing your stores, accusing you of selling unsustainable harvested fish, or having practices that don't meet their standards. So one of the things that these large organizations depend upon are these certification programs, and basically they're well-intentioned and they're attempting to be responsible and responsive, but at the same time it points out the conflict between some of these NGOs, governmental agencies, and the industry itself. And I think we need to keep that in mind when we talk about sustainability and when we talk about what we can do to help meet the global food security needs of the future. The NGOs have been an important force. They're important in, for instance, slavery issues, illegal and unlawful fishing, a whole lot of other child labor in the fishing industry and the global fishing industry and a lot of things, and they do a lot of good things. In our experience, they've also been an impediment to adopting a technology that we think is environmentally sustainable, and I'll talk a little bit about that. This is AquAdvantage salmon. Even our opponents show this slide. Those fish are the same age. They're sisters. They came from the same breeding, the same spawn. Fish in the foreground weighs about 300 grams. Fish in the background weighs over a kilo. They're about 11 months old. That's the AquAdvantage salmon. You don't get a bigger fish, it just grows faster. If you look at the growth curve for the young fish, you can see that the growth rate in the early life phase is 4 to 6 times greater than the non-transgenic control. These fish are genetically identical except for the presence of the AquAdvantage gene construct. In later life, you can see the growth rates begin to normalize, but you still get that 4 to 6 kilo fish significantly before you harvest a non-transgenic fish. And the opportunity there is a more rapidly growing fish that reaches the same terminal body weight, but it's a more efficient fish, both in terms of growth and in utilization of nutrients, as I'll show you. What is AquAdvantage? It's a very simple construct. I'm not going to show you a lot of data, but it's basically the Chinook growth hormone gene. The cDNA from the Chinook salmon, which is a Pacific salmon, which codes for the growth hormone. That's the same growth hormone that exists in the Atlantic salmon that's under the control of a different promoter. So we put a fish gene in a fish, and it's a fish gene that produces a protein that consumers already eat and have been eating for as long as we've been eating salmon. That construct was put into developing embryos, and the embryos, when the young fish hatched, we screened for fish that were, first of all, had taken up the construct and fish that were expressing the construct. Expression of the construct was illustrated very quickly in that rapid growth phenotype. When you have a mixture of fish that are not expressing and expressing in a tank, it's very easy to pick out the expressors. because at, for instance, 4 to 6 weeks, they're 2 to 3 times the size of the other fish in the tank. We've been breeding these fish, and another common misconception is that somehow we inject these fish every generation. Once we establish the founder line, we've been breeding those fish for over 25 years in our hatchery in Prince Edward Island. We're now on the 12th generation of AquAdvantage salmon by perfectly conventional, typical conventional breeding methods. How do we produce AquAdvantage? We knew when we were developing this fish, and this is where I came in 2006, that first of all, although there was support within the industry and in a lot of areas, particularly in the biotech area, that this was an interesting opportunity. But there were also a lot of people that were concerned about potential environmental impact or effect on biodiversity or effect on wild populations. So one of the things that we did was we took a look at the fish and said, we think we can grow this fish in a basically contained aquaculture system. A closed system where it can be land-based, it can be physically sequestered and isolated from other species, and the growth rates are fast enough that we can do that economically. So that's point one. The second aspect is we can produce this fish as a sterile triploid fish. You heard a little bit about triploids with Jack's watermelon this morning. We don't have to irradiate. We have something simple called high pressure. If you put the eggs at a certain stage of development under high pressure, you can basically cause them to become triploid. They have 3 sets of chromosomes instead of 2. Triploid animals can't breed. They're physically sterile, as you heard about the watermelon. So what we do is we use something called a neomale, which is pretty neat too. There are a lot of fisheries people here, but basically neomales are genetic females that can produce sperm. You can do really neat things in fish. So the genetic girls can produce sperm, and you can use that sperm. We use a homozygous neomale, 2 copies of the gene, to fertilize regular wild-type Atlantic salmon eggs. What you get then is a progeny that has a single copy of the gene. You can then render that egg triploid, and you can assay that in a fluorescence-activated cell sorter, another high-tech instrument found in medical laboratories and in most biotech laboratories. And so we can certify before every release of every batch of eggs that those eggs are greater than 95% triploid. Actually, our batches run greater than 99%, but the FDA only required us to be 95% or more. So this fish is literally equivalent to the production of a high-quality, highly purified pharmaceutical product. This is the first time in history that these sorts of standards have been applied to a living organism. And to my knowledge, the only existing example today. In 2015, the FDA, after a long review, public meetings, some of the people in the audience, Lisa Wettig, for instance, spoke at the public meeting in 2010 when the FDA released their initial review, they finally approved the product. This is the first genetically engineered animal approved by the FDA. The product is approved for 1 hatchery and 1 grow-out site, which happens to be in Panama, with an opportunity for adding additional sites using a process called a supplemental new animal drug application. There's a long list of post-approval reporting requirements. Like a drug, we're subject to inspections. We're subject to adverse event reporting. We're subject to quarterly reporting of operation of our fisheries and so forth and so on. But the important part is that we were approved. When they approved our fish, the first thing that the FDA said, which brought a chuckle to most of the people in the audience, was that it's an Atlantic salmon. We knew that. They looked at the 2-dimensional gel electrophoresis and concluded that it really was an Atlantic salmon. But it met all the criteria for Atlantic salmon. Most importantly, and without going all of the details in this very busy slide, there are no significant differences in composition, behavior, or any other measurable element between AquAdvantage salmon and wild-type salmon. There are some differences. Some of those differences are due to triploidy. They're not really relevant in terms of either the health or safety of the animal or the composition of its flesh. But they contain the same sort of nutrients in the same ratios and the same percentage and the same high-quality protein as other Atlantic salmon. And they don't represent any greater allergic challenge than eating salmon. If you're allergic to salmon or finfish today, you would be allergic. You would likely be allergic to AquAdvantage. It's not different than other fish. But if you're not allergic, the likelihood of having a reaction specifically because it's AquAdvantage is virtually nil. Concluding that food from AquAdvantage salmon is as safe to eat as food from any other Atlantic salmon. This is all available, and the citation is there on the FDA's website. In the phenotypic characterization, we basically measured everything that you can measure in a salmon. And for those of you who've done clinical studies and GLP studies, when you're working in a well-characterized environment, let's say human health, you know what the normal serological profile for every metabolite, every blood cell, every measurable analyte in a human is. Those numbers don't exist at least they don't exist in any credible, usable way for fish, even like Atlantic salmon, as popular as that is. So we had to do a lot of controls. We did commercial controls, we purchased fish, we purchased samples from grocery stores, and we did all of the comparisons with farmed, otherwise acquired, and our AquAdvantage. We, of course, had to do our controls, which were the AquAdvantage with the gene, and the non-transgenic controls, which were genetically equivalent, but without the gene. Last but not least, but perhaps most significantly, when they concluded the environmental assessment, there's a delightful term that most of you are familiar with. It's called a FONSI. Growing up with Happy Days as a teenager, FONSI was always but it's a finding of no significant impact. Our fish are raised in contained facilities. They're physically sterile. They're not permitted to be in sea cages. They basically have zero interaction with wild populations. The FDA and National Marine Fisheries and NOAA also signed off on the environmental assessment, concluded that as we promised to produce and grow the fish, that this represented no significant impact on the environment. In Canada, the story is much the same. Interestingly enough, and I don't say this with any either embarrassment or acrimony, the Canadian system in some regards is better. Canada has a sort of a dual system called a new substance notification regulation, which requires notification. Let's say you're producing a novel organism. You report to Environment Canada the nature of the construct, all the details, the same sort of information that we provided to the FDA. And under the new substance notification, there's a research exemption, but there comes a time when they'll actually do an evaluation and determine whether this represents an environmental risk. It's the equivalent of the environmental assessment in the US. And we went through that process, and the Canadians held an independent outside peer review, as well as their own review, and concluded that we represented no risk to the environment, the same conclusion that the FDA had. They actually reached their conclusion in 2013, 2 years before the US released their results. The other part of the Canadian system is the novel foods regulation. In Canada, they have a lovely law called novel foods. And Health Canada, Environment Canada, and the Canadian Food Inspection Agency review and look at the same data that the US FDA looks at. They looked at the composition, they looked at the allergenicity, they looked at all the food safety, everything that you could measure in a fish. And basically they concluded that this was, after all, an Atlantic salmon and represented no special threat to Canadian consumers, and therefore they had no further questions. So it wasn't really so much an approval process, but it was more analogous to our GRAS process in the US, saying, it's a food, guys, you can eat it. Neither country required any special labeling as a consequence of the AquAdvantage gene construct. So, we now have two major countries, Environment Canada, Health Canada, and I just talked about their process, who've concluded that this is not a big deal. It's a fish that grows faster, but if it's grown properly, is equivalent to the traditional food. What are some of the implications of this? This is a paper that was published in 2015 and talks a little bit about the nutritional requirements and performance. If you look at feed consumption or feed intake of transgenic salmon and non-transgenic controls, our AquAdvantage ate basically 3 times more feed than the non-transgenic in the same time period. Not surprising. They're ravenous. They literally live to eat. Also not surprising, the thermal growth coefficient was 3 times higher than the non-transgenic controls. If you feed them, they will grow. If you feed them 3 times as much, they grow 3 times as fast. We already knew they grew fast, but this was nice data showing that irrespective, and there are two, I should mention there are 2 groups here. One group is a plant-based diet, which has been used and replaces 50% of the fish meal. The other is a traditional commercial diet, which has the normal fish meal component. Regardless of diet, regardless of ploidy, the transgenics ate 3 times as much and grew 3 times as fast. The feed conversion rates, and this is important, this is the efficiency of taking the food that they consume and turning it into useful protein, was about 20% lower in AquAdvantage salmon. Now again, this isn't surprising. In most growth hormone supplemented animals, terrestrial animals, that have been studied, and there have been a lot, in general they show an improved feed conversion. But it was reassuring to see this. In terms of growing salmon in land-based systems, having a 20% feed conversion benefit is a huge benefit. And the next slide shows an even more interesting observation. The protein retention, and protein is one of the most expensive components of the diet, was about 5% greater in the transgenic fish than in the non-transgenic fish. Protein being the most expensive component, a 5% benefit and a 20% benefit in feed conversion means that this fish can be grown at a lot lower cost, and it grows a lot faster than the traditional food. Lipid retentions were also higher in the AquAdvantage than in the non-transgenics. So, if you can grow this fish, and you can grow it in land-based systems, and I've been telling people here that you could virtually grow it anywhere, let's hypothetically say that we put a facility somewhere in the continental U.S. And let's just arbitrarily say that we put it within 500 miles of a major city. If you're transporting fish from Oslo, 3,200 nautical miles, or from Santiago to Miami, 3,600 nautical miles, you're going to use, your carbon footprint is going to be about 25 times higher from either of those locations than if you're trucking that 500 miles to that metropolitan city in the United States. More importantly, the cost of that transportation, or equally important, I should say, it can be about $2 per kilo to fly that salmon from those European or Chilean destinations to the U.S., and it's about 10 cents a kilo to truck it from an American site to an American city. The cost benefit is significant, and the saving in carbon footprint is enormous. If you look at production model, and this is just arbitrary, models are models, they're good or they're bad, they're as good as the input in. If you just assume a 10% improvement in feed conversion, I haven't given us the full 20%, but if you assume a 10%, the cost of production, including transportation for AquAdvantage, is below a conventional salmon grown in that system by almost 60 cents, and is below the cost of production in sea cages and imported into the United States. So you have an economic benefit that you probably don't even need, because producing it even at the same cost or slightly higher cost, you still have a huge benefit in that transportation component. So these sorts of systems are big warehouses with fancy aquariums in. The tanks can be very large with a variety of designs. Modern RAS facility has a hatchery, has a sort of a juvenile area where you keep the young fish, usually called smolts, until they get up to 100 grams or larger. And then it has the larger tanks, and these can be of a variety of configurations. But these are recirculating aquaculture systems where more than 95% of the water is actually recirculated, reused, purified and reused. So the net release of water is only the water that you lose by evaporation or for cleaning of some of the drum filters and some of the equipment. If you look at the opportunities, and these are non-exclusive alternatives, I would never suggest that sea cages will go away. They will always be a part of salmon aquaculture for countries that have beautiful coastal fjords and cold water, and where salmon aquaculture is a religion, as it is in Norway. It's not just an industry. But it is a sustainable industry, and well managed can provide a very high quality product. But if you look at the sea cage opportunity, you have a lower initial capital cost, not free, but you do have a lower capital cost. You do need that long coastline, preferably with inlets and protected areas to try to keep the weather away from your net pens. But you've got pathogens, escape of fish, predators, sharks, seals, use of medications, fouling of coastal waterways, biosecurity issues. Anyone who knows how to spell ISA knows what that can happen to a geographical production area. You have exposed working conditions and a whole lot of issues before you even get to transportation, which can be as much as $2 per kilo. And that's at low oil costs. When the oil costs go higher, and they will, it'll go up, be higher than that. Wrasse, on the other hand, are expensive to build. 1,000 metric tons, roughly $15 million. 6,000 metric tons, roughly $90 million. But if you have a rapidly growing fish, and you can grow a rapidly growing fish that is more efficient, you have a better feed conversion, better nitrogen retention, you have greater biosecurity. Your fish are basically insulated from the environment. You don't need to medicate them. You've reduced the opportunity for any interaction with any wild species. You've reduced the transportation costs. You've cut the carbon footprint by a factor of 25. You've basically assured that there's a reduced environmental impact. There is never a zero environmental impact of anything, including standing here in front of you today. But you've reduced it to a manageable level. There is a higher capital cost, and depending on where you site it, if you have low-cost energy, these can be very, very competitive. If you have high-cost electricity, they become less competitive. I mentioned we would talk about wild-caught. Wild-caught can be managed. They're highly subsidized by the federal government, if you read the newspapers or the trade papers. When the Alaskans overfish their stocks, they go to the government for emergency funding to help the fishermen. When they have a bumper crop, they go to the government to buy the surplus to keep the prices up. Anytime they have a problem, they go to Washington, and Washington gives them $10 or $15 million to basically keep them supported, whatever else happens. It's a nice industry. I like Atlantic salmon. They've been a thorn in our side, or I like Pacific salmon. I do like Atlantic salmon. But the Pacific salmon industry, I think, we represent an opportunity to help them save their fisheries. And I really wish that we could get them to understand and accept that. We will compete with the Norwegians, we'll compete with the Chileans, we'll compete with the Canadians, we will not compete with the Alaskans. Last but not least, they do something called stock enhancement and ranching there. No one knows what the genetics of those fish are, and they release 2 billion young fish every year. And they catch some of them 3 or 4 years later when they mature. I don't know whether it's a sustainable fishery. It has the opportunity to be sustainable. I think some in NOAA think it's sustainable, and I hope they're right. But at the end of the day, if you look at the economic benefits, the social benefits, and the environmental benefits of land-based recirculating aquaculture, and I haven't gotten to economic benefits, job creation, and reduced dependence on foreign imports, land-based aquaculture with genetically improved stocks make a great deal of sense. And I think meet the standards of the 1992 World Summit on sustainability. Last but not least, let me make a pitch. This is not just about growth. Growth happens to be the first and the easiest endpoint to do. And we've developed other rapidly growing lines. We hope to have other products after this long and arduous regulatory process that we've endured. But the real opportunity, think of a world without infectious salmon anemia, without kidney disease, without some of the other rickettsial diseases, for instance, that have plagued the Chileans. The use of molecular genetics and molecular biology to genetically introduce resistance to major pathogens represents a huge opportunity. It would be literally a huge boost to the global salmon industry if you could free them from the burden of these. And anytime that you have large numbers of animals at high densities in a single location, you're going to be subject to those sorts of diseases. You can move that over to shrimp, white spot, Toro syndrome, and so forth. Molecular biology and genetics offer the opportunity to insulate global production from those sorts of plagues of pestilence that occur periodically and wipe out entire geographic areas of production. So you can improve disease resistance, growth and performance, nutritional characteristics, flesh characteristics, and last but not least, as we've done, you can do reproduction, control of fertility, and we have more elegant controls of fertility in development where you can assure single-sex populations, monosex populations of fish that are incapable of reproduction, which would be huge in any species, not just salmon, also trout, pick a species where you need to control fertility. So that's my story. That's been a long time happening, but NADA 141452 is now a reality. And at the end of the day, it's really about food. So thank you very much for your attention.
I put my glasses on so I can see you. So you were talking about growing the fish in near a major metropolitan area, and I'm wondering right now with regards to the environmental assessment that was done specifically for growing out in tanks in Panama. What does that mean? I mean, if you want to have this happen, what are going to be the next steps to have it be able to grow somewhere else besides Panama?
As I mentioned, the process, as was discussed in the 2010 VMAC meeting, would be what's called a supplemental new animal drug application. There's an analogy. Let's say that, for lack of a better term, I'm producing a pharmaceutical product. Pick one, doesn't matter what it is, cholesterol-lowering agent. I'm producing it at a factory in Kalamazoo. I want to open a factory, or I want to have a contract facility in Reno, Nevada. You basically file an application with the FDA describing the facility, describing the environmental assessment for the surrounding areas and so forth and so on, and you make that application. It is generally approved within 6 months or so. What would be in that application would be a description of the facility, the management practices, all the considerations, whether it's in a floodplain, what the seismic activity, temperatures, all those sorts of things, all the physical characteristics, proximity to waterways and so forth. So in principle, the process would be fairly straightforward. We'd file a supplement. We'd file an environmental assessment as a part of that supplement, describing the site completely, and the FDA would inspect the site prior to approval. But if they found it adequate and satisfactory and meeting their standards, they could approve that site, and then you would have another approved manufacturing site. When I talk about the Canadian and U.S., in Canada, we can practice what we do anywhere in Canada because they've said it's okay. We have no questions. As long as they do it the same way, they can do this in 50 locations or 5 locations. In the U.S., it's site by site. We're regulated under the Food, Drug, and Cosmetic Act. We're basically a living animal regulated as a drug. We're not complaining because we ask for this, but at the end of the day, it doesn't make a great deal of sense. But we're more than willing to work with the system, and we think we can get this done. It's a good question, though.
You briefly mentioned about disease-free fish. Can you expand a little bit on what is the molecular approach that you are following for disease-free fish?
Well, disease-free is a term that has a temporal component. You're disease-free at any moment in time. You're disease-free until you're not. But when I talk about disease resistance, for instance, I'll use the example. At the Rowett, they've developed poultry that are resistant in transmitting avian influenza. The birds die that become infected, but they can't transmit the virus to other fish. That's as good as a cure, because if you can't transmit it, you lose one bird, not a whole poultry house. If you lose one fish, that's not a big deal. If you lose 50 million, that's a big problem. So I think, by analogy, we think that there are opportunities for viral diseases to introduce viral resistance at a genetic level for diseases like ISA, and perhaps even for diseases like rickettsial bacterial infections. Disease-free does not mean there will not be biosecurity. There's always a need for biosecurity. We have exhaustive biosecurity. That's a whole other talk. We practice the belt and suspenders approach. Our hatchery is certified as disease-free, but I can tell you disease-free has a temporal component. The idea of introducing specific resistance to specific pathogens is something that I think is very attractive, because it will help insulate the industry from these catastrophic tragedies that occur periodically. Does that answer your question? I don't know. It doesn't sound like it.
I was intrigued by the diet issue where you said they eat 3 times more and you get 3 times more growth. If you look at the issue which Jude Capper talks a lot about of maintenance energy, you should actually do better because you've got fewer days of maintenance energy, and therefore the proportion of growth in a faster growing animal should actually be faster. I realize you're not giving detailed numbers, but in fact, do you see that benefit?
The nutritionists play with those sorts of calculations now, and you raise a good point. We don't know enough at this point other than we know that we have a better feed conversion and we certainly have a faster growth rate. We're just beginning to scratch the surface. That data I showed you is between 0 and 300 grams, by the way. We're now looking at later stages. Part of the problem that we have in running these studies is the AquAdvantage grows so much faster than the controls that the AquAdvantage are at 2 kilos when the controls are still at 300 grams, and you have to bring those fish all the way through, which takes another 18 months. So there's a lot that we have to learn about the nutrition and the performance, other than on a very simple level, they're a lot more efficient, and they certainly grow a lot faster. There's a lot to be known there. We're also working with soybean growers and other groups looking at novel feeds, trying to optimize the feed for AquAdvantage. That's an area of emerging research, I think. Go ahead.
My question is more from a marketing side. We've all heard of happy cows. Would you say your fish is a happy salmon?
We have very happy fish.
I personally think it's not a happy salmon because you have deprived it of sexual satisfaction. So I would have second thoughts about eating your fish. Because I take a lot of care when I grow my sturgeons. I make sure they're happy. I put males and females together, and then I delay satisfaction. Do all sorts of things to make them happy before I put them to harvest the caviar. So it's a serious marketing issue for us. We make sure that our consumers are very sophisticated. So make sure they know that there are caviars coming from happy fish. So how would you market your fish in those terms? Some people may feel guilty that they're eating a fish that's been deprived of its.
In practice, what we've heard, and I need to talk to my German Shorthair too, because we had him neutered when he was a year old. He may be having some reservations. In all seriousness, when we did this development, we had outside veterinarians come in, and we looked at behavior, predator avoidance, swimming in water column. When you take visitors to our hatchery, these fish are like pets. They're hand-fed to satiety every day. If you go to Panama, when you appear at the tank, they're a lot like your aquarium fish. When they see that shadow, they know it's time to eat. By all measures, the behavior, the performance, all of the other things, they don't do predator avoidance very well. They're much more interested in eating. The other thing that we found, we have no problem, a big problem in the salmon industry is early maturation. Early maturation is males which mature early and then try to breed with females. We don't have that problem because we have all females and they're triploid. So we seem to have sort of happy eunuchs, if you want to stay with your analogy. But the fish give every indication of doing very well. And we've had now more than 25 years experience growing these fish. So when the proof of the pudding is always when people come in and see the fish. They're expecting to see, and I once used this in an interview with a journal. A journalist asked me a question, and I said, you have to understand, these fish grow faster, they don't get bigger. And then, without thinking, I said, you don't get a salmon the size of a Buick. What do you think he printed? He printed, you don't get a salmon the size of a Buick. And I learned, and I can say that here, and I can say it in jest because it's a few years ago, but it reaches the same terminal body weight. It just gets there faster, and it gets there on less feed. So, that's, if you're in the business of growing salmon, those are pretty interesting economics. Yes, Tyra.
I'm actually more interested in the happiness of the consumer, and we won't get into sexual habits or anything here. But there was a book back in the 70s about, making the perfect tomato called Hard Times, Hard Tomatoes. And I've watched the poultry industry go to these fast-growing meat machines. And I know the meat's changed. My wife talks about it, and I noticed it to some extent, but she notices the way it cooks a lot of things. Mm-hmm. how exactly does the meat turn out? And have you carefully scrutinized how the meat changes as you grow it 3 times faster?
Well, the only thing I can say, and I presented some of this data back at the VMAT conference in 2010, we sent our fish to test kitchens of some of the largest retailers and restaurants, seafood restaurants in the United States. They were included in blind taste tests with salmon sourced from Canada, Norway, and Chile. And in 3 separate blind taste tests in their test kitchens, we tested number 1 or number 2. They looked, and these were professional chefs preparing 3 different presentations and tasted by professional tasters. They looked at things like flesh quality, odor, appearance, firmness, basically the kind of things that foodies and chefs would be concerned about. We presented that data. It's on the FDA website. When we presented it, one of the FDA lawyers said, well, maybe we have to put a label on it that says your salmon tastes better. And at the time I said, we'd be really happy if we didn't have any label, because we think it's just that our fish was maybe a little fresher than the fish that they source from some of those other countries. That's a long answer to say we have not seen any difference. You can make a bad tasting salmon. If you harvest a salmon, don't bleed it out and don't chill it overnight, and you put it on your barbecue, I don't care where it came from. It can be king salmon from New Zealand. It's going to be chewy. It's going to be tough. It's going to be, it's not going to have that flavor that you're looking for. But properly prepared, properly harvested, our fish is equivalent, and in fact, in those taste tests, superior to the competitors. We've seen no differences. We have a local semi-famous chef in Canada that lives a few doors down from our hatchery. He actually uses it in his culinary school to teach his student chefs to prepare salmon dishes. And some of the hot maple smoked salmon he produces is among some of the best salmon that I've ever had. So it's a nice commercial, but take my word for it. When you have the opportunity to buy it, take it home and try it. I think you'll like it.
I have a quick question. I'm from Louisiana, and triploidy in oysters is, I would say, almost common.
Or
now.
tetraploidy. Yeah.
Tetraploidy. Is there a correlation to that? I mean, that's something that I know that on a small scale, at least in Louisiana, but I think it's through up and down the seaboard. I mean, that's common practice, is it not, in oysters? And why could
it's one of the many mysteries. And Jack talked this morning about the seedless watermelon. You can produce a seedless watermelon with a metaphase blocker like colchicine and generate triploids that way, and it's not regulated. You can irradiate seeds, and that's not regulated. You can mutagenize, and you can do family selection, and that's not regulated. You make a very precise gene, and this goes to our conversation last night about CRISPR. You make a very precise change in a gene which hasn't changed in more than 25 years. We can prove the same sequence today that was isolated in the first fish, and some people say, this is a big deal. It's one gene, and it's a gene that they've been eating forever. So it's a part of that larger discussion with consumers about what is the impact. A good friend of mine, Max Holtzman, the former senior advisor to Tom Vilsack at USDA, told me once over breakfast, because we were talking about this, and it was before our approval, and I was saying, Max, I just don't understand it. This is really good stuff. He said, Ron, American consumers don't want to hear 2 words in the same sentence, technology and food. And he was right. So one of the things, and Jack mentioned it this morning, if you look at our website, we talk about food, we talk about sustainability, we talk about opportunities, we talk about American productivity. We don't talk about what promoters we use and how we express the gene and so forth, because that's completely lost. And frankly, science has never won the game for us. It doesn't matter whether it's a Monsanto or the newest startup company. And our discussion last night will bear that out, I think, as well.
But this is oysters. I mean, it's the same, pretty much the same gene. I mean, that's the same genre.
I will wager if you and I go out, and let's go downtown Raleigh tonight, and let's just stand in the street corner and say, will you eat tetraploid oysters? We won't find anybody that will go near them. And yet they've been eating them for
Let me know which part of you.
Thank you very much. Okay, our second presenter is Glenn Cor-